without further ado, I would like to invite Brian Zimmerman, who is the Director of Conservation Science at the Bristol Zoological Society, and Charles Fusari, who is the Director/Curator of the Tropical Aquarium of Paris. So I'm going to now stop sharing my screen. And Brian, if you'd like to share your presentation.
Great. Thanks, Mark. Can you all see that?
Yes, that's perfect. It's full screen.
Okay, marvelous. Good evening, everybody. Thanks for the introduction, Mark. And you get a double bill tonight. Both Charles and I will be speaking. I'm going to have the first half where I'll be talking about the inception of a conservation project for freshwater species in Madagascar. Some of you may have read recently, there was a big news release about how freshwater fishes are among the most threatened vertebrates on the planet and are obviously probably more neglected than many of the other species like mammals and birds. So hopefully this presentation tonight will enlighten you all and talk a little bit about something, a species that you haven't heard of before. So this is In Search of the Red Girl. First of all, a little bit of a taxonomy lesson. So who is the red girl? I don't know if there's any taxonomists out there, but I do find this slightly interesting, and it's informative when we're talking about this species, just to let you know. It is a freshwater fish in the order of Ptychochromis and in the family Cichlidae, and that's an important factor that I'm going to come back to in just a little bit. The genus name for this fish is Ticochromis, and the species name is insolitus. And if you're a taxonomy or a linguist geek, you'll enjoy dissecting this scientific name. It's half Greek and it's half Latin. The Greek part means fold, and a fish, perhaps a perch. So going back to that taxonomy, the fact that this is in Perchidae is a good indication of where we're going from the taxonomic perspective. But also insolitus is the Latin side of it, and that means unusual, odd, or queer. And I'm going to come back to why that's important in a little bit too when we're talking about this fish. But it was a reference to somewhat atypical appearance of the species and the morphology of aquarium raised specimens that were used in the original description. Here's a picture of the fish, which is called Jubamina, or the red girl. So Jubamina is the local name in Madagascar, and it translates to red girl. And here's a picture of an adult specimen from London Zoo's aquarium from a few years ago. Now, the interesting thing about this is the ones that have red on them, which give them the name red girl, are actually the male fish. And these are two male fish from the aquarium in London Zoo. So they're not girls at all, they're boys. This is a girl, this is a female Jubamina, and you can see that she's got virtually no red at all on her fins, and she's a rather drab looking specimen. I mentioned that this species is a cichlid, and that's quite important. Those of you who've kept fish in aquariums at home are probably very familiar with cichlids as a family. They're very popular in aquariums for lots of different reasons. They're relatively straightforward to keep in most cases, but they have lots of characteristics that make them fascinating to look after. They're usually sexually dimorphic, so you can tell the difference between males and females. They're an egg-laying fish. And probably what's interesting about them is they practice parental care. So they look after their eggs and they look after their fry when they've hatched from the eggs. And in order to do this, they usually form very strong parental bonds, and they are territorial. So they look after the space, whether that's the area where they've laid their eggs in or where their fry are being looked after. They aggressively defend that from any intruders. And this behavior, this aggressive behavior, occasionally turns out in a kind of murderous way for particular fish that have invaded their territory. To talk more generally about the freshwater fishes of Madagascar, you can see on this bar graph, this shows just the endemic species of fish found in Madagascar. and this freshwater fish. You can see that the largest portion, or the family with the largest number of species in it is cichlids. There's 41. There's actually probably more than that, but at the time that this was produced, there were 41 species described or recognized, and that was followed up by rainbow fishes. And you can see that in some cases, there's only one species per family, but cichlids are the most prevalent family in Madagascar. And this species that we're talking about, the Jubamina, is found in one particular area in the north of Madagascar in the Ambobo River, which is a tributary of the Mangarahara River, which forms part of a very large basin called the Sophia River drainage. And this is in the western escarpment. So these are drainages that flow to the west, to the northwest in Madagascar, which is fairly unusual because most of the water in Madagascar is on the eastern side, and western Madagascar is significantly drier. But the place where this river, the Ambobo, originates is near the village of Maratondrano, which I'll talk a bit more about in a minute. The other common name for this species is the Mangarahara Cichlid, named after the Mangarahara River where it was first described from. There are 6 sort of main basins in Madagascar, and the area that this fish comes from is one of the northwestern basins, which the Sophia drainage is the primary drainage of this area. There are other areas and other basins in Madagascar, and typically the fish fauna from those areas is significantly different across each of the basins. So the situation back in 2004, which is sort of a year after this story kind of starts from my perspective, was that the species hadn't even been named yet. So you can see the IUCN listing calls a Ticochromis species Jubamina, the local name, and it was classified as critically endangered back in 2004. And this excerpt from the cichlid fishes of Madagascar says that it appears to be extremely rare within its known and restricted range in the Sophia drainage. The extent of occurrence and age of occupancy are estimated at less than 100 kilometers and less than 10 kilometers respectively. Despite targeted efforts in it, it is rarely collected, and given the high rate of deforestation in the region, construction of dams, and the introduction of exotic competitors and predators, the species faces a very high risk of extinction. So this is the reason it was classified as critically endangered back in 2004. But a year before that assessment, I had the privilege of receiving a group of these Jubamina at London Zoo. 19 juvenile individuals were donated to us. They were the offspring of some wild-caught individuals that were brought in by the ichthyologist Jean-Claude Noursat and given to 2 hobbyists, Sonia Gunan and Dave Turo, who were successful in breeding them once. And they gave London Zoo 19 juveniles to look after. And the intention here was that we would set up a new breeding program for the species. After we did some analysis on it, we realized that it probably wasn't suitable to develop a program based on these 19 individuals because they were all siblings. They were all from the same batch. So they were related. So that would instantly create this genetic bottleneck. Also, as a cichlid that's aggressive in territory holding, and they can get fairly large, they had large space requirements. And at the time, the exhibit space in London Zoo Aquarium didn't have very much room. And this factor of parental care that cichlids employ mean that there's a generally very high success and survival rate when they reproduce. And that's great if they're in the wild in terms of carrying on the next generation. But in an aquarium setting where there are few to no predators and the fish are looked after, that high survival rate can automatically create a saturation of the zoo population with the genes of just a few individuals. So ultimately, 10 years later, so after quite a bit of time, a number of those 19 individuals had eventually died off. They were either killed by their siblings or they died of other causes. And there were only these 2 males left in the aquarium at the zoo. Right at this time, Zurich Zoo decided to do a survey. They've got a number of projects in Madagascar, and they wanted to know how many different Malagasy fish species were being kept in European zoos and aquariums. And the aim was to establish suitable and stable captive breeding groups for key species, those that were most threatened. And then the idea is also obviously to share the husbandry and breeding knowledge for those institutions that were keeping and breeding those fish, so that we were doing it in the best way possible. With the genus Ticochromis, what the survey revealed is that 2 of the species, Oligocanthus and Grandadiri, were held in a few institutions and in significantly large numbers. Both of these species are classified as least concern, however, whereas the Ptychochromis insolitus was thought at this time potentially to be extinct in the wild, definitely critically endangered, and it was only held in 2 institutions. And you can see the numbers there are very small. It was 3 males and 1 female. So the 2 males were at London Zoo, and the pair, the single male and female, were at Berlin Zoo. So back in Madagascar, right around this time, it was a worrying situation. In fact, Nurasat and Duram reported that even back as far as 1999, they'd faced a distressing sight. As far as we could see, the bed of the Mangarahara River was completely dry and all life had disappeared over an unknown distance. With this information and knowing how few fish were held in aquariums, I rang my colleague Rainer Kaiser at Berlin Zoo and explained that he was our last hope of being able to breed these fish as far as we knew in Europe, and that he should do something about it. And he very diligently set up a breeding tank behind the scenes at Berlin Zoo Aquarium, specifically designed as a breeding and reproduction tank for this pair of Mangarahara cichlids that he had. And we had high hopes that he would get them to reproduce. However, because they're cichlids and they're territorial and they're aggressive with each other, it ended up resulting with the male killing the only known female, which was obviously quite distressing. So we decided to set up something called the fish appeal. And this was done in a bit of a light-hearted way through the press team at London Zoo. And we basically set out as a wanted poster, and it was a fish appeal that was sent out via press release. And we wanted to try and target hobbyists and fish breeders around the world, as well as other zoos and public aquariums outside of the European region. And the aim was to try and see if we could find if there were any females that we could pair up with these 3 males, the 2 in London and the 1 in Berlin. We set up a separate ZSL email address to see what form of interest we would get, and to make sure that my own inbox wasn't going to be overwhelmed with replies. And I was very glad that that happened, because we had a massive response. It was incredible because the media interest in this story was phenomenal. We reached 174 media outlets in over 100 countries, and an estimated reach of over 63 million people. And that doesn't include social media or international radio and TV broadcasts. That was just print. And you see some of these headlines here from the BBC and Metro referring to this fish as gorgeously ugly. And I think that was an actual slip up on my part when I was asked by the BBC reporter to describe for a radio audience what the fish looked like. And I'd said, they looked a bit like gargoyles. They're a bit grotesque. I said, but they are gorgeously ugly because they have these flowing fins. And the media picked up on that and used that as the term to describe them. And of course, everybody wanted to know what a gorgeously ugly fish looked like. So it generated a lot of interest. And that created thousands and thousands of emails sent to this fish appeal inbox. There were lots of people just sending well wishes, saying they wished us luck in finding a female. There was some hate mail, people that said, who cares, it's just a fish, it doesn't matter if they're the last ones. And there were some interesting suggestions from people. saying that we should try and clone them or freeze their DNA, which of course zoos do anyway. There was suggestions to crossbreed them with other species to save the genes. And of course, because it was a global reach, people from all over the world were writing in and saying that they knew where there was a fish. There was feedback from someone in Malawi that said there were lots and lots of cichlids in Lake Malawi. Of course, it's a different species. There were hobbyists from all over the world saying that they thought they could find them. There was someone in Venezuela that said they have a river outside their office and he was sure there were cichlids in there. Of course, it was a completely different species. And people from basically all over the world were giving us hope and generally false leads. Until on the 8th of June in 2013, there came a much more interesting response. And this is from a gentleman in Madagascar. And he said, following our investigation near Mangarahara in May last year in the river Mangarahara, we no longer find the fish, the Jobamina. But in the other river in the southwestern Mangarahara to the west side of Angivoni, near to Mangarahara, the fish is still found. He went on to say, it's about 45 to 50 kilometers from Mangarahara, but you have to use two means of transport, a 4x4 car and motorcycle, and there's a section of road impassable by car for about 35 kilometers, and the rest is on foot or by bike. So it's a very inaccessible place. However, this was enough to give us hope and mobilize support for an expedition to go and see if the fish indeed still existed anywhere alive in Madagascar. And we had logistical support from Toronto and Denver Zoo, as well as from the British ambassador in Madagascar, and all of the ZSL directors, and everybody contributed as much as they could financially to support an expedition. I then had to assemble a team of the right people, and this included the guy that sent us the first lead. So it's the guy in the picture here in the upper left of the slide, Guy Tam Hayak, a local Malagasy gentleman who's got a lot of interest in fish conservation in Madagascar. There were various team members from Toronto and London Zoo, as well as the university in Antananarivo, the ichthyologist team there, and one of the students at the university. I'd not been to Madagascar at this point, and although I'd done lots of field monitoring expeditions to other places around the world for freshwater fish, I knew that we needed to carry lots of equipment if we were going to do a proper search for the species, but I also wanted to travel light, thinking about that long stretch on foot at the end, and I didn't want to carry too many things. Nonetheless, our equipment list was extensive. We needed seine nets and dip nets and hand nets. We wanted to be able to measure the water quality of the area where the fish potentially lived, so we needed test kits and probes for that, as well as the calibrating fluids and sterile bottles to do that. We need to be able to find our way. So things like GPSs and torches were important, but also PPE, things like chest waders. If we've caught the fish, we wanted to be able to transport them potentially. So we had transport bags. We wanted to document everything, including the fish we caught, through photographs. And of course, we needed detailed maps and all the other things that you need when you're out in the field looking for fish. And of course, we needed to stay somewhere. So we had to bring our own tents and sleeping bags, and we didn't want to get sick. So we brought sterilizing tablets for water and anti-malarial tablets, of course, and first aid kits. When we arrived in Antananarivo, we went to probably one of the coolest shops. Anybody who's a map geek like I am, there's a great map shop there, and you can get pretty much every single square inch of the country on a ordnance survey map. And this was obviously a very valuable resource because we needed to identify not only where we were going, but also potential sources of water and springs that might hold the fish that we were looking for. We also went to the university to talk to the ichthyologists there to learn a bit more about what they were doing in terms of surveying Madagascar's different regions for fish, and to get some logistical tips and practical advice in terms of access. We planned for the trip to be 3 weeks in duration. We needed 2 vehicles and we had 7 team members. And obviously we needed sufficient time not only to get from one place to the next, but also enough time to meet with people to gain permission to access sites and to fish in the rivers. And also for the team to work together and learn how to fish cooperatively together and to know who is doing what task. I mentioned this is in the Sophia drainage, and our goal was to sample in several of the rivers and tributaries along the area where there was suitable habitat to ensure to find out whether or not the fish existed in other locations where it hadn't previously been found. And we wanted to take some water quality analysis to find out what the situation was in the wild in terms of if the habitat was degraded. Our intention was, although we were looking for the Jubamina, we wanted to record any and all sympatric species that were found in the same habitat that shared the same places to learn more about the ecology, the overall ecology of the rivers. And in order to learn a bit more about what was going on there, we wanted to find out if there was some direct action that could be taken to improve the conservation status of this species if we found it. So our first fishing spot after driving for a couple of days was in the village of Maravato. We had some reliable information that this area had a significant amount of water in one of the rivers near to the village. So we decided to stop there first. In order to fish in these areas, you've got to get permission from the locals, from the communities. And so here we went to the local village elders and had a lengthy discussion in their home about how we would access the site and if it would be okay if we caught fish and examined them. And they were happy with that. They really wanted to know our opinion about Britney Spears and Manchester United, 2 topics that I know absolutely nothing about, although they did have lots of posters around in their houses of those 2 subjects, which was quite interesting in the middle of nowhere. We also talked to them about gaining access, and we got some good advice from them, but that obviously didn't make things very much easier. For those of you who've been to Madagascar or places like it, you realize that some of the roads that are on the maps can scarcely be called roads, and they make interesting driving. And when we arrived at the first site in Maravato, the fishing site, we were very encouraged. There was water, which was a bonus, considering it was the dry season, but also the water looked fairly good in appearance. There was a significant amount of water. It had depth. It was clean and clear. So we were very hopeful of finding fish here. And we had a lot of help. So we spent the entire day using all the different techniques to work out what fish were living in this area. And we had a lot of help from the local community, who obviously fish in these areas on a regular basis. So here are the guys helping us with the nets. It was really, really good having an extra several dozen pairs of hands. So we were able to operate more efficiently. And we had a great audience of people very curious to what we were doing. And that was probably one of the most interesting things of having such a large number of people watching me while I was doing water quality. Whoever knew it was such an interesting job. I even had a duck, as you can see there, swimming away, keeping an eye on things. Unfortunately, this first site was very disappointing in terms of the catch. The only species we were able to catch was Oreochromis mozambicus, which is a tilapia, which is not from Madagascar. It's from Africa, the proper Africa continent. And it is widely introduced in many places around the world as a food fish because of aquaculture. Our next stop was going to be Antisakabari in the north. And this again was a site that seemed to hold some promise. Unfortunately, after driving for over a day and a half along this road, one of our key vehicles broke down and we lost a day there. And while the vehicle was being repaired, we trekked on foot along this road and realized that the road got worse and worse. In actual fact, it was virtually impassable even on foot. So we decided to abandon that idea and headed down to Mangarahara, which is the kind of local capital of the region. It's a fairly large town with quite a few people living there. And it's the gateway to the Mangarahara River. So not very far from Mandratsara, you can see the flowing Mangarahara River. Well, not during the dry season, because this is a picture and it basically substantiated what that report was from 1999, saying that during the dry season, the river is essentially not a river at all. There's just a few isolated pools. And this presented a problem, obviously, if you're a fish, but also for the local community, because they rely on the water. And this picture here shows that locals were doing their laundry in a dry riverbed, and in order to do that, they had to dig down 2 to 3 meters through the sediment in the riverbed to reach water. So it was an issue for them as well. And we came across some children and older people fishing in the river in the few pools of water, but we quickly learned that there were actually no fish there, and they were catching these tiny shrimps and water beetles and snails to eat instead. A demonstration of just how difficult the situation is for some of those communities. And so we decided to go for our big key area, which was Marotondrano, where we had the kind of accurate report that the fish still might exist in this area. And so we headed on according to the map, the straight road from Mandratsara to Marotondrano. But you can see this is the picture of the road. It wasn't exactly straight driving, but we eventually got there. And it was clear that Marotondrano was a slightly different community compared to some of the others. And the fact that there was already significant amount of development aid spent there, and particularly looking at water resource management. And that was for the rice farming of the local community. So when we got there, we decamped to a local restaurant and we discussed our plan, and we discussed with the local community about access. And this included with permission from the national park. And within about 15 minutes, one of the local fishermen came up and gave me Jubamina. Unfortunately, it was dead and dried out. And although this was not exactly the result I was looking for, it did fill me with additional hope, because this was a fish that was recently dead. And so I thought there's a good chance that there are some still alive somewhere out there. And when the locals realized that I was on the hunt for fish, they brought me more and more. Unfortunately, they were all dried out ones, and I developed a very large plate of interesting variety of endemic Malagasy fish, but all dead. And I eventually explained to them that I was interested in these same fish, but in a live version. And then I was immediately whisked off down a trail towards the place where they were used to catching them. So after a short hike, we ended up in this place, which is the Ambobo River, but it's not flowing during the dry season. It forms these temporary pools. And to the right of this picture, you can see these sticks placed in the water. Those are put into the water by the local community to help them to catch the fish, to kind of act as a barrier or a restraining net. They set the area with these traps, and when we went down there, they went and quickly retrieved one of the traps and emptied the contents into a bucket. And you can imagine my anticipation after being in the field at this point for over 2 weeks. Would we actually see one? And I was very excited and surprised that we finally had a living, breathing Jubamina. And in actual fact, we ended up seeing several on that first day. They caught a number of young individuals, as well as a few other of the native species found in this river. So it was very exciting, and we were all very, very thrilled with this. And of course, we celebrated that evening and sat down and talked about, what was going to be our next step now that we knew that the fish was still extant in the river. What was also exciting for us is that it wasn't just the Jubamina that was there, but there were a number of other endemic fish found in the same habitat. You had the species in the hand here, Salvagella robusta. It's probably one of the most delicate fish I've ever worked with. You just look at them the wrong way and they seem to die. but also there were other cichlids there. The bottom middle picture is a Melancisca. It's a Paratropolis gymnopria percularis, found in this area. The orange fish on the bottom right is Paratropolis neurosati. They get bright orange in the breeding season. And the top right is a type of killifish called Pachypanchax, not yet described to species level, called Sophia. And the other one in the top middle is a species called Rheocles durami. Charles is going to talk a little bit more about that in his half. And this was the only specimen of this fish, critically endangered, that we caught on the expedition, and it's not been seen since. There weren't just endemic native species there, though unfortunately there were tilapia present and even goldfish in some of the fishing areas that we looked at. And this is obviously a huge issue for freshwater fishes when an alien invasive species becomes dominant and competes for the limited available food sources. So that was a concern. And there were other things that concerned us quite a bit. There was a lot of fishing and a lot of extraction of fish from the area. There was also a lot of extraction of water for rice farming. A lot of water was being pumped away and diverted to rice fields. And worryingly, they were using this local plant, this thing in the middle picture at the bottom. It looks a bit like a type of yucca, which has poisonous sap in it. And they would use this, they would extract the sap and use it to poison the pools of water and kill all the fish in there. Madagascar has lots of other environmental problems. Lots of the area, particularly around here, is burned, and the forest is regularly burned seasonally. And it often gets out of control. This burning then leads to serious erosion when the rains come, and it burns the toxic ash that's left over and a lot of exposed ground as sediment. And hence you get these situations in the river where large numbers of huge volumes of sediment build up and basically choke the river. So based on all of these findings, we realized we couldn't just celebrate that we found the fish and walk away and go back to the UK and say job done. And we decided that we needed to execute an emergency response. So our plan was to capture 20 individuals of each of the endemic species in an equal sex ratio of 10 males and 10 females, pack each one in an individual breather bag, which is a special bag for moving fish around, and transport them to our partner Guy's fish farm in Andapa in order to have a kind of insurance population and hopefully get them to breed and build up insurance populations of the fish at his place. So we then spent the next several days catching fish, and each one had to be measured, photographed, and then transported overland back to the village by foot in order to repack them. We also did detailed water quality analysis, so we had a good record of the physical chemical needs of the fish, and we recorded all the habitat characteristics, including things like substrate and what types of invertebrates and plants were found in the water. So our fishing results yielded not quite 20, but close to it. Jubamina 19, but 11 males and 8 females. We also caught 21 of the Paratropolis neurosati, and only 9 of the Melancisca. We caught 23 of that killifish, the Pachypanchax. And then once we caught them all and packed them up, we drove overland for 31 hours from Marotendrana to Andapa, which was not a fun ride. We drove almost continuously, only stopping to refuel. And despite that long distance and the challenges of the road, we only had 1 mortality, which was pretty impressive given the extreme temperatures we were driving in as well. And we got there and we released them in these ponds here. Guy's looking into one of the ponds that the fish were put into. And within 2 years of doing this, we were very, very pleasantly surprised because we had over 12,000 Jubamina that were hatched in Andapa, as well as thousands of other species, the Lamina and Pachypanchax. The only one that we didn't have success was the Melancisca, I think because of the small numbers and perhaps maybe the ponds weren't suitable for their needs. It was a good thing too, because back at London Zoo, around this time in the same year, both of the males, or surviving males, had died within a month of each other. And although this was a really sad occurrence, because those two individuals had obviously developed a huge fan base from within the zoo and outside, all was not lost, because the specimens went up to the National Museum of Scotland, and they formed part of their collection there. I'm now going to hand over to Charles, who's going to talk about the next stages in the project and where we're at and where we're going. So thanks for your attention. I'm going to stop sharing my screen.
Thank you very much indeed, Brian.
All right, hi everybody. I'm gonna stop sharing my screen. Is that fine now? Yeah.
You're on. There you go. That's perfect.
All right. Hi. I'm really pleased to be here tonight. So at the moment, I am the new director of the Tropical Aquarium in Paris since March 2021. So it's very, very new. And before that, basically, I was working in London Zoo since 2015. First as the deputy team leader of the aquarium, then the team leader, and then finally I was able to be the curator of fishes for the Zoological Society of London. And I think one of the reasons why Brian hired me at the time was maybe because I was speaking French, and this was a good thing, because he wanted me to get involved in this project in Madagascar, to be able to really ease the communication, with the partners on site, because they were all like speaking French, and it was pretty difficult with the communication between them and London Zoo, and so I've been very lucky to be involved in this project, and I did my first expedition in 2016, which was like 3 years after the first expedition. So the goal, so in 2016, we got some fund from the European Union of aquarium curators to be able to go back to Madagascar. And one of the main goals was to familiarize myself with the project, with the species, with the sites, the different partner, and the country itself. And then we wanted to go back to the Mboa River as well to try to capture more Paratrophus gymnopercularis, which is the Melemsisca, because we only had 9 individuals in the ponds in Ndapa, and they weren't breeding successfully. So we wanted to get more of the species to really have a significant brood stock. And then we wanted to go back as well to the ponds in Ndapa, where all the fish were breeding, to check on the different population, and as well to improve the welfare and the breeding success. And we wanted as well to train our partner there, who managed the ponds, in water quality analysis. So when we arrived in Madagascar, we obviously spent a few days in the capital and Tananarivo to do the route planning with the partners, to meet with our partners at the university, and to do the official visits with the British ambassador. And then we went to we set off to go back along the Mboa River in Marotondrano, where we spent, unfortunately, we didn't have that much time, and we spent only 1 day along the Mboa River to try to sample and catch more Melemsisca. Unfortunately, we didn't catch any of the species. Maybe it was the fishing technique, maybe it was the sites, maybe it was the fact that we haven't had the time to do multiple days along the river. But the good news was that we still found the Jobamena. We still found as well the Lamena, the Paratrophus noricity, and we caught as well a native species of goby. Then we went to see the ponds in Ndapa, where our fish were breeding since 2013. And the 2 first ponds that you see at the bottom of the slides are the ponds where our fish are. And this video here, I hope everybody can see the video, is a pair of Jobamena. The small one is the female, and she's like guarding the nest, and you can see that they clear the patch of the mud, and they have these rocks where they can lay their eggs. So the female here is going to lay some eggs, and then after we can see the male here, she's laying some eggs, and then we can see the male slightly bigger, like fertilizing the eggs. They're going to do it another time, so I'll let you appreciate the species. That's it. And here you can see a female Jovamina guarding the nest after, and it was interesting to see as well that once the male fertilizes the eggs, then he leaves the nest, and it's only the female that guards the nest. This is the other species, the bright orange species, which is the lamena. When they breed, they have this really nice color on them. And what's nice about and what's very special about this fish that is they lay their eggs in caves, and you can see here the male fertilizing the eggs, and the female here is smaller at the entrance of the nest. So all those videos have been captured in the ponds in Ndapa. And this is the third species that breeds very well in those ponds, which is the species of Pachypenchax that hasn't been described yet. And you see that there's loads of different individuals, and they like spending their times between the marginal plants where they lay the eggs. Very, very curious little fish as well. So these are the recommendations that we made for the pond management. We recommended to cure the ponds and get rid of as much as possible of the mud at the bottom of the ponds, and add lots of different rocks and different shape, and create caves to be able to stimulate the egg laying for the Pterochromis nerisati and for Ptychochromis insolitus as well. And for the Pachypenchax Sophia, we advise to increase the marginal plant cover on shallower depths to create sheltered habitats, and let the air algae grow for them to be able to lay their eggs. And then this is our partner Guy that we trained in water quality analysis that will carry out every month, and then it will then inform us in this regular report that is sending us regularly. So then I went back in 2017 for an expedition, and here the idea, really the goal of the expedition, was to walk the whole length of the river and try to assess the area of occupancy of the species within the Amboaboa River, try to list all the different threats and map the different barriers, whether they're natural barriers like rapids or waterfall, or anthropic barriers like dams. And we wanted as well to assess the river's health using macroinvertebrate samplings, and assess as well the feasibility of setting up community ponds that will be managed by the local community to farm the endemic fish. And this idea was to create a source of livelihood for the local community, but as well create insurance population for the local species endemic to the Amboaboa River. So this is the map of the river. See if I can use the pointer, the laser pointer. Maybe not. Anyway, so this is the map of the river. At the bottom right corner, it's the protected is the national park of Marotondrano, where the headwaters of the rivers are located, which is a very very positive thing for us, which means the headwaters of the rivers are protected, and the river is still flowing. And then the little there's 5 little marks along the river on the very first third of the river, which represent the dams, and the light green areas along the rivers are the rice fields, and all those dams basically are diverting the water towards those rice fields. So I mentioned that the headwaters were located within the national park. So this is a video of the headwaters within the national park of Marotondrano. So the water is still flowing a lot, and there's a very dense forest which helps to protect the river. And this is the national park managed by Madagascar National Park. It's the park of Marotondrano. So when we carry on along the river, you see that the color change here in the color coding of the river, which means like where it's blue, it means the water is flowing, and where it's in yellow, basically, it's when the water isn't flowing anymore. And you can see that there's on the picture on the side, there's only intermittent pools, which isn't suitable really for the species. And if I go back the slides before here, what we found in terms of the area of occupancy of the species, it starts really the species are only found from the town of Marotondrano. I don't know if you can see because the map is a bit on the side, and then stopped where the water stops flowing. So in terms of the whole length of the river, the species are only found in like only 1/4 of the river, because at the top, at the bottom, on the last part of the river, there's no more water, so it's not suitable, and we don't find the species. And on the top part of the river, there's natural barriers that prevents the migration of the species upstream, so that the species isn't found there. And this is the rest of the river, basically, which is completely dry during the dry season until it reaches the Mangarahara River at the end of the map. In terms of the list of threats, we noticed, so there's obviously lots of deforestation, and this is a picture that has been made at the outskirts of the national park. So there's more and more deforestation going on, unfortunately. And as Brian explained earlier, when you cut, so the forest cover and the vegetation, it leaves the soil completely bare. And during the rainy season, the soil is being washed away and end up in the riverbed. And this is a video I made along the river as well, where you can see the thick layer of sediment. So this is the Amboaboa River. This is the local fishermen that we use when we do our sampling session to try to catch the fish. And I'm like sitting on the sand in the middle of the river, and there's this stick poking out of the sediment that I'm on the lift, and you can see that. So for information, I'm like 6'3", and I'm going to hold the stick that was completely buried in the sediment, like really high above me. So there's approximately 2.3, 2.5, like 3 meters of sediments accumulated in the river. Next slide. So the other major threat was the water diversion. So as I described before, there's a series of dams along the river that are used to divert the water to extensive rice fields. So this one is one of them. We can see the channel as well in the picture at the bottom that divert the water to the rice paddies. And this is a video of the main dams in Marotondrano. You're going to see so the channel where the water is diverted, and here where there's and then you can see as well that is going back to the river. So this is diverted towards the rice fields. And then when I'm going to turn, you can see there's the little trickle of water here going back to the river. So Brian mentioned the alien species, or what sometimes called the invasive species as well. So we sampled 13 sites along the river, and here's the results in terms of fish we caught. The species in red are the endemic species that are endemic to the Amboaboa River. Species in green are the species that are native to Madagascar, but not especially endemic to the river. And then the species in black are the alien species, and you can see that the native and endemic species are only found in a very mainly between point 8 and 12 of the river, while the tilapia species is being found across the whole river and nearly across the whole site. The fishing activity that Brian mentioned as well. This is just after the village of Marotondrano. There's a very important fishing activity because we are very near the village. and the villagers really built those kind of barriers made of twigs and branches that blocked the whole width of the river. And on this picture, you can see that there's one just behind the other one, so it's very, very intensive and leaves really no chance for the fish to escape. It's very unselective as well. They catch every kind of size, which isn't great. So it means like the fish that are in age to breed are captured as well, and the juveniles are captured, and I haven't had the chance to grow and breed to renew the population. In terms of assessment of the river's health, we use macroinvertebrate sampling to really understand what is the health of the river, and the result that we found is confirmed what we observed. Basically, the site 1 is very close to the protected area where the health is good, and then while you go down towards the village of Marotondrono, where there's lots of water abstraction and lots of fishing activities, which is towards sites 4 and 5, then you've got a very poor health of the river. And then after it picks up a bit more, and then in site 14, it's where the river is completely dry, basically. And then one of the main ideas we wanted to do as well in 2017 is assess the feasibility of starting to build like community ponds in the village of Marotondrono along the river, so the local community could farm the native, the endemic fish, and create insurance population. So we met with like a few farmers and people from the local community that had ponds already, and we talked to them about this idea, and it was the first kind of contact we had with them, until we went back in 2018 to really start digging more ponds and helping them to catch the local fish. So I went back in 2018, and so we still wanted to do a plot sampling along the river, to check on the fish health population. We selected only 6 sites that were located in the part where the river is still flowing. We wanted to establish the partner with the Madagascar National Park that managed the protected area, for them to do monthly water quality monitoring. We trained their rangers to do water quality analysis along the river, and we set up the Pond Owner Association, and we started to build ponds as well. So these are the 6 sites that we selected. The first one is the one closer to the national park, and the number 6 is the site that is just before the river starts to dry, to run completely dry during the dry season. These are some of the specimens we caught. So we caught all the specimens all the native species that were present in 2013, except the Rheoclis de rami, and I'll come back to the species a bit later. So we sat down with the Madagascar National Park and established a partnership for them to gather water quality analysis data along the river on the 6 sites monthly, and report those findings to them, so we can use them after in further research. So we trained the rangers, and on the table you see on the side is what they've been gathering since 2018. So now we've got like across the 6 sites, we've got more than a year and a half worth of data, which is very, very, very valuable for us and for what we try to do with the fish. This is a little video about the Pond Owner Association that we start. So here's the Mboabwa River, and you can see the rice fields on the side. We gave lots of tools and materials for the local community to help them to dig the ponds. There were some ponds that were ready already from 2017. So we caught a few fish we introduced in the first pond. And in 2019, unfortunately, I haven't been able to go back to Madagascar because my wife was expecting a baby. She was 8 months pregnant, and although I was very enthusiastic and I thought I would be able to go back, she really told me that it wasn't really a good idea to go to Madagascar in a very remote location when she was 8 months pregnant. So I followed her advice and I stayed at home. And the only thing we've done in 2019 was for a partner, Guy, that managed the ponds in Ndapa, is really taking a lead on the Pond Owner Association in Marotandrano. So he went back there to check on the ponds that were being built by the local community and train the local community as well in pond management. And as a result, he trained 23 people from this local community. There's 22 ponds that have been built along the river. And from this first animals that we fished, that we introduced in 2018, we got 830 fry that have been produced. And those fries have been split and dispatched in the other ponds that have been built since. In 2020, unfortunately, was a bit of a weird year for everybody. So due to the travel restriction because of the pandemic, I haven't been able to go back. So I kind of took advantage of the situation to create a website that really talk about the project and about what we're trying to achieve. So if you have a chance and you want to know more about the project, you can go to fishnetmadagascar.com. So what's next? In 2019, we applied for a National Geographic grant, and we got the money, which was really amazing. And this grant is administered by the Bristol Zoological Society. And this is what we're trying to achieve with the grant. We've got a 2-year funding that is split in 4 different areas. The first one is we're trying to achieve a translocation feasibility study, which means like we want to try to take the species where they're found near the village of Marotandrano and where there's lots of threats, and we want to like move some of the specimen upstream to the within the boundaries of the national park where they will be protected and where they can establish a population here. So we're looking at the feasibility of that, whether the species will survive within the boundaries of the park. And this is where the water quality data that those ranges of Madagascar national parks are very useful, because they allow us to understand whether the parameter where the fish are living at the moment in the river, whether they are the same parameter that on the habitat host when we want to put them within the boundaries of the park. And we are doing as well, in collaboration with ZSL and the Institute of Zoology, a disease risk analysis. This is to look at whether there's any health risk for those fish that we want to move from where they're located to the national park. Whether there's risk for the fish themselves, health risk, but whether there's as well health risk for the habitat host. We're still working on insurance population, so with this project of community ponds along the Amboa River. In terms of like the population ex situ within the zoological institution, we are in a very better situation that we were back in 2013, where we had only 3 males left in zoos. Now we've got thousands and thousands of Jobamena across different kind of zoos and aquariums across Europe that have been bred in the ponds in Ndapa, and that have been exported as well after to Toronto, then dispatched all those fish within zoos and aquariums. And then we're working on the community engagement program with an environmental NGO from Madagascar, and their part of the job is to really try to conduct a perception study with the local communities and understand how they use the river, how they perceive the environment, and what do they understand about the fish population within the river. And we're hoping that this will help us really to target or to like really to do some tailor-made conservation action in collaboration with the communities along the river. And this perception study as well would gather information about how the dams along the river are being managed as well. And obviously, we will carry on going if the restrictions allows. We will carry on going back to Madagascar to like monitor the habitat and the population within the river. What's next? We've got a lot to do, especially in terms of ex situ breeding research on the Joba mena, really to understand the influence of the seasonal variation on the life cycle. And this will help us really to know whether we can move the fish upstream and whether this fish will thrive and will be able to breed within the boundaries of the national park. We want to try to breed the Malagasy in the ponds because we haven't been able to breed the species due to the lack of individuals within the broodstock. Something as well we would like to do, because there's no population of Paratropus nerisati within zoological institution, we would like to establish this species within the zoo and aquarium community to set up backup and insurance population. One of the species that have been caught in 2013 was the Riocles de Rami, which is endemic to Madagascar and maybe as well endemic to the Mboaboa River. But since then, when we went back every year, we never really caught this fish again. So since 2013, we caught only one individuals, despite maybe between 30 and 40 sampling sessions along the river. This species is classified as critically endangered as well. So we're very concerned about the species, and we're not very sure whether it still thrive or whether it can be found still within the Mboaboa River. So what we want to do is go back and do what we call the eDNA monitoring that has been really used in previous projects in Greece that Brian has been involved, which the principle basically is to try to take some sample of water along the river and filter it, get all the different biological material within the water, extract the DNA from this water and compare it with a database and see whether you can find the DNA of the fish floating in the water, which will tell us whether it's still present in the river or not. So this we're trying to raise some money to conduct this eDNA monitoring project. We would like to sample as well and explore nearby rivers to have the bigger picture and see whether we can find the Joba mena, the Malagasy, and the Lamena in those nearby rivers that we haven't been able to explore because they're quite remote. And that's it, actually. That's it for what is next. Obviously, all those projects are short-term to medium-term projects, and as a long-term project, there's a lot of reflection going on how we can really improve, the health of the river in long term to ensure really the population of fish to thrive within the Mboaboa River. And this is the acknowledgement to all the people that contributed financially or logistically to the different expedition and to the project so far. I don't know if Brian wants to add something to this.
No, I don't have anything extra to add, but yeah, just acknowledging all of the partners and supporters that have helped with this project.
Charles and Brian, thank you very much indeed. That was really fantastic, full of intrigue and also some really hopeful messages. So we're a little bit over time, but I think we should still have time for a few questions. Is that okay, Charles and Brian?
Yeah, that's fine.
So if anyone wishes to ask a question, you can type in the chat if you want to. If not, I've got questions too. So in that case, perhaps I'll just kick us off with the first question, which is, so you sampled one river in an area where there are lots of inaccessible rivers. Would you expect to perhaps find other Malagasy endemic species in other remote river systems throughout the country, or do you really think that this is a unique river system?
Shall I take that, Charles?
Yeah, go ahead.
That's a really good question, Mark. And I would expect to see and find many new species, probably many of them undescribed. I think the exploration of Madagascar's fresh waters is not complete by any means. And a big part of that is down to access in some of these remote areas, which is probably the only thing that's saving the fish that are there because they're remote. But we do know that the anthropogenic changes happen very, very quickly. And in those places where there is community access and the water can be diverted for whatever purposes, the fish are usually the first things to go. So yeah, I would expect there to be a lot of other species still yet undescribed. But the group of animals in the Amboabo River, I think to me represents a kind of relic of what Madagascar used to have. And the only reason, as Charles, I think, pointed out, that those fish are still there is because of that Marochandrano National Park, which protects the headwaters and keeps the river flowing, at least partially.
Thank you very much indeed. Do you want me to read out the questions from the chat, or do you want to read them for yourselves? I don't mind either way. I'm happy to read them if you wish.
Yeah.
Yep, so a question from Benjamin Shaw for either Brian or Charles. It sounds like there was an emotional appreciation of Jubamina in the UK from the London Zoo campaign. Were the attitudes of the Malagasy people towards this fish any different? Did they appreciate the species more as a food resource?
I can answer a bit that if you want. I don't think the local communities saw the fish here very in the same way as the as we saw it in the UK after the campaign. I think they just really see the fish as the food source and nothing more. And I think most of them are, or maybe all of them, really didn't really know that this fish is maybe only found in this river across the whole planet. So I don't think they had the same perception that we had, I think.
Thank you very much, Charles. Another question from Alan Goldsmith. The original fish were from hobbyists. Is there any role for hobbyists in the conservation / captive breeding of any of these? I know some Malagasy cichlids are kept and bred by hobbyists and a few of the killifish as well. But is there any formal connection, current or planned, with specialist aquarist societies?
I can answer that one. So the quick answer is yes, there is. The regional collection plan for freshwater teleosts was recently published, in fact, at the beginning of this year. And part of that maps out how we will collaborate both within institutions within the European Association of Zoos and Aquariums, but also harvesting that information and that knowledge and the skill base within the hobbyist sector. It's obviously not easy to do that because you're talking about individuals versus institutions. However, we do know that there's a lot of really good information and skills out there. So we already are, for a number of other species, working with the hobbyist sector and the specialist breeders. So there's no difference here with the Malagasy species. And in fact, in North America, there's a formalized program that uses the hobby sector for breeding, for conservation.
Thank you very much, Brian. As we're a little bit over time, I'll maybe leave the questions for now, but we've had some lovely comments for people thanking you for fascinating presentations. And I would also like to reiterate, again, thank you both for a really wonderful presentation. Thank you everybody for joining us this evening.